Okay, so finally, our last topic focuses on an equally interesting topic. It's on using psychoplastogens to selectively rewire pathological neural circuits. And this area of research is being pioneered by Dr. David Olson and by his collaborator, Mark Russ. So I'll introduce both of them now. So come on up to the stage. Thank you. David Olson is an associate professor in the Department of Chemistry and the Department of Biochemistry and Molecular Medicine. Professor Olson's expertise spans central nervous system medicinal chemistry, molecular and cellular neurobiology, and behavioral pharmacology. His academic lab discovered that psychedelics promote structural and functional neuroplasticity in the cerebral cortex. They coined the term psychoplastogen to describe small molecules that produce rapid and long-lasting psychedelic and ketamine-like effects on neuronal structure. And they invented the first non-hallucinogenic psychoplastogens that are capable of producing sustained therapeutic effects in preclinical models after a single dose. In 2019, Dr. Olson co-founded Psychedelics Therapeutics, a company developing non-hallucinogenic psychoplastogens for treating brain disorders such as depression, post-traumatic stress disorder, addiction, and Alzheimer's disease. He is an associate editor at ACS Chemical Neuroscience, and he's received several awards, including the Geordie Folk Pye Award from the American Society for Neurochemistry, the Sigma Xi Young Investigator Award, the Life Young Investigator Award, and the UC Davis Chancellor's Innovator of the Year Award in 2021, among many others. We're also delighted to have Mark Russ joining our conversation. Mark Russ is the Chief Executive Officer for Psychedelics Therapeutics, located in Massachusetts. Psychedelics is pioneering efforts to understand how reversing cortical atrophy and rewiring pathological neural circuits can drive rapid and sustained healing for people that are suffering from many psychiatric and neurologic disorders. Mark is a globally experienced life science executive who most recently served as an entrepreneur in residence and advisor at Atlas Venture. Prior to Atlas, Mark served as Group Vice President of Neuroscience at Shire Pharmaceuticals. And over the course of 13 years, he led teams across the United States, Europe, Canada, and Japan at multiple clinical and commercial stages. He was nominated by Pharmaceutical Executive Magazine in 2017 as one of the top 11 emerging leaders in the life sciences. So we are very excited to have both of you join us for this last part of our panel today. Okay, so let's start with neuroplasticity. We've heard a lot about this topic. So many of us, as you've heard, grew up thinking that our brains are hardwired. So as we grow up, they're plastic, and then you hit some stage during adulthood when you can no longer rewire your brain. But as we've heard about today, we are beginning to understand in neuroscience that this is not as true as we thought it was before. So your research program really taps into targeting plasticity to selectively rewire circuits that are damaged in brain disorders. So David, can you start by describing the contribution of neural circuit dysfunction to disease and what brain disorders this might relate to?
Sure. So it's important to remember that activity in specific circuits controls behavior. And in many brain disorders, there's a physical change in the brain, particularly in the so-called stress-related neuropsychiatric diseases. These are things like depression, post-traumatic stress disorder, and substance use disorder. And there's a part of the brain that sits right about here. It's called the prefrontal cortex. It projects to a whole bunch of other brain regions that control mood, motivation, fear, reward, cognition. And so you can imagine if there's dysfunction in the prefrontal cortex, you can have a range, it can cause a range of disorders. And in many of these cases, including the neuropsychiatric diseases, but also neurodegenerative diseases like Alzheimer's disease and others, the prefrontal cortex physically atrophies and kind of shrivels up. And so what we were really trying to do is to use a pharmacological agent to rapidly regrow these neurons so that they can communicate more effectively with other parts of the brain and alleviate the symptoms of these diseases.
Great. So can you define neuroplasticity for us and how it works?
So neuroplasticity is a very broad term, and if you ask 10 neuroscientists what it means, you get 10 different answers. And so the kind of neuroplasticity that I'm really referring to here is typically called structural neuroplasticity, the physical changes in the structure of neurons. And those changes in structural plasticity can often lead to functional changes, how those how those neurons actually communicate with other partners. And so when we're talking about the effects of a psychoplastogen, we're really talking about the physical rewiring of the brain.
Okay, terrific. So David, you and other labs have revealed that psychedelics and related compounds are exceptionally good at promoting structural and functional neuroplasticity in cortical neurons. So can you tell us about how this discovery came about?
Well, it was a long time coming, I'll say that.
Yeah.
I'd say the beginnings of this came when I was a postdoc at the Broad Institute, the Stanley Center for Psychiatric Research at the Broad Institute at MIT. And this was a time in neuropsychiatry where there had been kind of a dearth of innovation. And everyone was getting really excited about this molecule called ketamine. And ketamine is an old molecule. It was been used by people as a dissociative anesthetic for many, many years. It's also known as Special K. It's a drug of abuse. But psychiatrists found that ketamine could really, really rapidly produce antidepressant effects. And now today, ketamine is an FDA-approved treatment for depression. At the time, when I was a postdoc, it wasn't FDA approved, but we were all very excited about the mechanism of ketamine. And it seemed that ketamine had this effect on cortical neurons to rapidly regrow these neurons. Now, something about ketamine that really stood out to me was the fact that it seemed to work across indications. It worked for depression. It also worked for PTSD. There was some indication that it could work for substance use disorder. And so ketamine is far from a perfect drug. And so when I started my independent career at UC Davis, I was really looking for molecules that could be better than ketamine, but produce ketamine-like effects on neuronal structure. And that's what really led me to the psychedelics. There was really rich literature, dating back to the 70s, clinical data suggesting that molecules like psilocybin and LSD might work for a lot of the same disorders that ketamine works for. Alcohol use disorder, depression, PTSD. And so the first question that we wanted to address was, do psychedelics produce ketamine-like effects on cortical neurons? And if so, could they offer some advantages over ketamine in terms of their circuit-level selectivity? Could they be more specific and avoid some of the other issues associated with ketamine?
Okay, so you're talking about the use of psychedelics for potential therapies. So Mark, in thinking about some of the ways that drugs are developed and used, what are some of the downsides of using traditional psychedelics in this respect?
Yeah, thank you. Great question, Dr. McAllister. And before I start, can I just say one thing? This is the first time I've been to UC Davis. I went for a run this morning along that arboretum. You guys have a beautiful campus here. This is a really, really nice place. You're very fortunate to have it, and walked around last night. So kudos and thank you for having me. The promise of early stage research and the promise of what's discovered, I think it was Dr. Moxon who so aptly said, do not underestimate, you cannot underestimate or overstate the challenge with which actually bringing a therapeutic, a neurotechnology, or any other type of invention to human use, especially in neuroscience, can be. And psychedelics are no different. So the promise that was so prevalent, and particularly since the early 2000s in the research David referred to with ketamine that eventually resulted in an approved medication, Spravato, opened the doors, but also opened the doors to many more questions about the ultimate scalability. And the simplest way I'll describe it from a scalability perspective is think of scalability from the earliest of stages. So that's things like IP. Many psychedelics that offered promise are either naturally derived products or ones for which there is no possible or potential IP. And there's different schools of thoughts on whether that's a good or bad thing, but it's nevertheless a complex thing to navigate. Manufacturing, API, active pharmaceutical ingredients. These are very difficult challenges to actually physically make either plant-derived, botanicals, all the way through to synthetic psychedelics. And there was, while there was promise, not a lot of clear answers on that. A host of other early stage challenges I could list, and then think about a second category at the latest stages, commercial stages, human stages. The way I tend to think about these things in the pharma space or the biotech space are called the 5 Ps. How can you solve for the 5 Ps? First and most importantly, patience, right? Is all this work, is all this money, is all this time and effort actually going to lead to a meaningful patient benefit? That's clearly the most important thing. Secondly, payers. Who's going to pay for whatever the innovation is? Again, that's something we've heard other speakers refer to and cannot be underestimated. Third, physicians, prescribers, or any other practitioners, if it's not a prescribing-based treatment. How convenient, how complex, how accessible, how much time is required? In the psychedelic space, many of these promising treatments require inpatient treatments. So many of you may have read about or heard of the promise of psilocybin, magic mushrooms, or LSD, or ketamine, as David referred to. 8 hours in inpatient, 5 hours, 4 hours, time to onboard, time to offboard. These aren't bad things. Lots of good patient outcomes can come from that effort, but scaling it, very, very difficult. Fourth, pharmacists. So dispensation, how do you actually get, in this case, a therapeutic to a patient? And at-home delivery, at-home access, medicine cabinet is something that is very promising and obviously makes a difference in terms of adherence, something that first-generation psychedelics, at this stage at least, cannot do. And then fifth and finally, the 5th P that I think about, policymakers. How accessible can a potential new treatment be in the policy realm, and what can it offer to enhance other policy aims? So first generation psychedelics, the likes of which ketamine is one example, but think of all the others, clearly an exciting space. Clearly we're rooting them on because we think they can and will help many patients if and when proved and approved by the FDA and around the world for therapy. But scalability, major question. And that's where Delics began to come in.
Well, and so that led to addressing some of these issues that you bring up for the traditional psychedelics has led to a second and third generation approach. So could you tell us a little bit about those?
Sure. The simplest way to describe it is first generation would be anything kind of plant-derived, natural, okay, everyone put up your hand if you've, no, just kidding. So the first generation psychedelics were really the ones that formed the foundation for this movement. A lot of mission-oriented researchers, investors, and others who a lot of us are standing on the shoulders of after 20, 30 years of hard work to progress the research, progress in the destigmatization efforts, et cetera, but essentially unmodified from their natural state. The second generation are therapeutics that companies have formed around to attempt to solve for some type of IP differentiation, as well as their PK/PD profiles, so their pharmacokinetic profiles. Can you find a way to perhaps lower what is traditionally an 8-hour experience to a 4-hour experience, or a 3-hour, or a 6-hour? In some cases, how can you lengthen them to make them more intense for those for whom the disassociative, mystical, or spiritual experience are deemed to be part of the therapeutic benefit? How can you formulate them in a different way to be ingested or cross the blood-brain barrier in a more effective way? So these are tweaks to natural first generation psychedelics, but they're steps, but perhaps not leaps. Third, and what we like to believe are final generation psychoplastogens offer a completely different approach, one that decouples the dissociative, hallucinatory, and other elements as well. We're often known as the company that's taking the fun out of the fungi, but there's actually a lot of other very critical, important elements that you need to think about. 5-HT2B agonism, for example. There are on some chemical scaffolds particularly serious risks, cardiac risks. The ibogaine scaffold, for example, is one in which people have died, taking cardiac treatment. Taking ibogaine treatments in Mexico, abroad, for various serious, very important reasons. Military veterans, many of whom I've spoken to, gained a lot of benefit, but you need an actual cardiologist with you, during these treatments to ensure it's safe. We're not only removing the dissociative elements, we hope to improve the tolerability for a lot of these second generation psychedelics as well.
Awesome. So David, I have a question about how your approach and your goal in developing psychoplastogens to treat psychiatric disorders, how does that compare to the treatments that are used right now that are focused on altering the levels or the function of individual neurotransmitters?
Yeah, so the most common medicines used for depression are things like selective serotonin reuptake inhibitors. Probably the most famous is something like Prozac. Mm-hmm. And the way that these molecules were believed to work for many, many years is that they block the reuptake of these key neurotransmitters like serotonin, and that results in very high levels of serotonin in the synaptic cleft. And people thought that rectifying this chemical imbalance might do something for depression. But that really kind of led to the so-called monoamine hypothesis of depression. But there was an issue with it. The issue is that things like SSRIs drastically increase serotonin levels immediately. But it takes weeks to months before patients experience any kind of benefit. And so after years and years of really hard research from people across the country and across the world, what we've now realized is that things like SSRIs seem to promote neuroplasticity. They're just pretty bad at it. They do it slowly over time. And so if you track that neuronal growth phenotype that I talked about in the cortex over weeks, the course of weeks to months with an SSRI, you'll see that patients get better at about the same time that their neurons regrow, not the same time that their serotonin levels go up. And so that again has led to the so-called neurotrophin hypothesis of depression, which again comes back to this idea of neuroplasticity. Now, the difference between something like an SSRI and a psychoplastogen is kind of the speed at which these work. SSRIs promote neuroplasticity through kind of an indirect mechanism that takes a long time. Psychoplastogens do this more directly, and within 24 hours can regrow some of these neurons to produce long-lasting changes in behavior. And for me, this really represents a paradigm shift in the way we think about treating these types of disorders. Previously, patients would be taking things like SSRIs every single day. And daily administration of any kind of therapeutic is going to come with a host of side effects. For SSRIs, it's things like weight gain, sexual dysfunction, etc. And that can, really decrease the tolerability of those types of medicines. But if you can think about using a small molecule as a catalyst for the brain to physically heal itself, now maybe you have to take the medicine once a week, once a month, once a year. That's unclear, probably will depend on the specific molecule and the specific indication, but it's not the same paradigm. It's not the daily administration of something like an SSRI.
So I am incredibly impressed with your synthesis of information and your innovative forward thinking of how you might be able to address this problem, and then your fearlessness at jumping right into the commercial world and developing a company. So why don't we have a video that I think we're going to show about your company. It's very short, and then I'm going to ask some questions about how you as an academic faculty member or student or trainee address this brave new world that you're doing very successfully. So let's watch that video if we can.
The prefrontal cortex, or PFC, is a brain region that controls mood, cognition, joy, fear, and more. It helps make us who we are. In many neuropsychiatric diseases, neurons in the PFC atrophy or wither. Branches retract, synapses are lost, leading to impaired cognition, disrupted mood, and other symptoms. Psychoplastogens are molecules that spark regrowth of key neurons, healing damaged neural circuits. By physically changing the structures of neurons in the PFC, psychoplastogens hold the promise of rewiring the brain to heal the mind.
It's a fantastic video, so please visit their website and take a look at that. It's very inspirational. So in thinking about your discoveries in your lab and how you can translate that into developing a company. Can you tell me what inspired you to do this, and then how you found each other and how this company got launched?
Honestly, I wouldn't say I was inspired to start a company. I would say that I feel a moral obligation as an academic that if we make a discovery that we think can really help people, we owe it to the world to try to bring that to patients so that people can actually benefit from it. And so that's the real reason I started a company. I got into science to be an academic. I'm not into business really. And so I guess that's why I tried to do this. And the way I was able to do it was by surrounding myself with people that are much smarter than me. And I think it's really important to know what you don't know. And for me, I felt like, on the scientific side of things, we have a really interesting idea, but to really make it successful, to do what Mark was talking about and translate this, to make it commercially viable, I had to really enlist the help of other people that understood that world better than I did. And so to do that, I relied on a bunch of resources. So I was very fortunate early on in my career to get one of these STARE grants from Venture Catalyst. And anyone else in the room that are applying for this, I'd really highly encourage you to apply for this program. It's great. As part of that, I went to an entrepreneurship academy. That really helped me to start building my network, to start talking to people and to see what does it take to build a company? What are patients and payers eventually going to be looking for? How can I convince other people to get on board and to help me with this? And I would say I started that process maybe back in 2016. And it really wasn't until I had talked to hundreds of people really looking for a good business partner that could help me achieve these goals. And then I met my co-founder, Nick Haft. And Nick had a very similar vision as I did. It was clear that he was quite bright and very well connected. And that's when we decided to ultimately start the company.
It's really, really fascinating, and I think inspirational to a lot of the trainees here who were thinking in a more entrepreneurship approach to research. So, Mark, from a company's perspective, how does industry see opportunities like this to partner with researchers at a university?
So companies, whether they're at the scale of Shire, when I was leading a team there of about 2,000, we had about 3 billion in annual turnover in sales, and a mix of other offices and presence around the world. From that scale, all the way to the VC world, where I was after, all the way through to small startup companies like Psychedelics now that are growing quickly. In all three cases, there is someone there who's scouring the academic landscape at any point, or working with third parties who specialize in understanding and taking snapshots of the landscape in whatever particular field of therapeutic interest they have, I would say on an annual basis. So the first thing you need to understand is there are people out there looking all the time. So that's the first piece of good news. The second piece is that the fragmentation of in-house R&D at vertically integrated pharma companies into more disparate, opportunistic, horizontally integrated search and development plays in biotech, VC, and elsewhere has really ossified over the last 10 years in a good way. So it's a long way of saying people like acquiring innovation, not trying to build it all in-house. So there is a large appetite. The challenge I'd say everyone needs to be cognizant about is that there can be a perception or misperception that it can be difficult to work on some occasions with tech transfer offices. And in my experience, I can't speak highly enough about UC Davis, the experience I've had here, and never had any prior to working with David and the team here, but had good corroboration from some other colleagues of mine who'd done some work in the past with UC Davis as well. So you guys should know that at least in the N of one, there was a good reputation out there. But there are other faculties, there are other universities that I've worked with in the past that if someone asked me today, would you work with them? I'd say, I'd think twice. I think, is this really, really worth it because of some of the challenges inherent in partnering with them? And I think the simplest way to distill that to you and how to think about things, if you are interested in building a company, if you are interested in scaling to grow, is think about any relationship you have, any licensing deal you make, any partnership whatsoever, as a starting line and not a finishing line. Okay? What I mean by that is, in cases that I've seen or have seen in others where there may have been challenges of partnering with universities, it's where an agreement, a license, a tech transfer, whatever it may be, was viewed as a final, okay, great, we did it, we made the deal, now you go off, company, you go and make something of it and check the agreement every time you need to see what to do when you have to cross the bridge on a decision, as opposed to the companies, as opposed to the teams who truly view it as a long-term relationship, who view it as the beginning of a journey together in research, in development, in relationship building, and reputation building, and all those things, and truly view it as the beginning of a long-term relationship. I hope that makes sense, but that's the simplest way I can describe the difference I've seen in the past. What I also want to, I can't overstate, is in all you heard from David's example of how he formulated and brought forward the concept that eventually became Psychedelics, there was a lot of self-starting in that. There was a lot, that entrepreneurship boot camp, the relationships he built, correct me if I'm wrong, David, but no one like came to you one day and was like, hey, you should go to this, you should do this, you independently thought of this, you took a step forward and you did it. And in my life at least, the times in which I've stretched to do something, or more importantly, didn't stretch to do something, were cases where I felt something seemed unattainable, right? Like, oh, I can't learn that particular skill, or I can't learn how to do that. That's for those people out there. They have something different than me. I don't know how to acquire, like, that just doesn't seem like something I could do. And the truth is, we all can do all these things, but there's no substitute for the hard work and the relationship building necessary to get there. And that, quite frankly, can take a while. It can be hard work. It's extra, but I don't want to overstate the amount of hard work that I know David has done and will continue, has continued to do to help bring this to fruition. So starting line, not finishing line.
Yeah, and I just want to emphasize something that Mark said. Starting a company is just the first step in the journey. And if you want to make this company successful, you got to be present and contributing every step of the way.
I'll also just add one thing there, ego, or how the humility. So, David spoke about surrounding himself with people who had skills or had done things that he didn't have, and assuming you can't know or don't know everything. That's no different for me. It's no different for anyone on our team. And that, I think, is one of the secrets of Psychedelics' success so far. We've been very fortunate to be able to attract some of the best and brightest talent who are all share this long-term mission, this long-term vision of doing something different in mental health, to actually bring a healing-based approach to a lot of these neuropsychiatric and eventually neurodegenerative conditions over time.
This is terrific. I have a lot more questions on here, but we're running out of time. I know that we all wish you both incredible success, because we will all benefit from the new therapeutics that you develop. So thank you so much for your hard work.
Thank you.